Welcome to this video on the biology of reward deficiency. We will explore how gene-environment interactions shape our dopamine and norepinephrine baselines. Our biological blueprint starts with genetics. While certain polymorphisms can make neurotransmitter systems less efficient, genetics alone rarely cause reward deficiency. They primarily create a physiological vulnerability. Key genetic variations include the COMT gene, which can clear neurotransmitters too quickly, DRD2, DRD4 receptor variations, which limit signal reception, and overactive DAT1 transporters, which remove dopamine prematurely. The environmental catalyst is often early life stress. Childhood trauma or chronic adversity activates the HPA axis, flooding the developing brain with cortisol and creating long-term neurological impacts. Through epigenetics, chronic stress alters gene expression. Methylation can turn off genes needed for dopamine synthesis, while simultaneously turning on genes that increase reuptake, further depleting neurotransmitter levels. Continuous exposure to cortisol becomes neurotoxic to the developing reward pathway. In response, the brain downregulates dopamine receptors, essentially dulling its own reward and arousal centers to cope. This combination of genetics and environment results in a structurally blunted system. Individuals may experience chronic fatigue, apathy, and anhedonia, leaving them feeling persistently understimulated. This chronic deficit creates an intense subconscious drive to fix the neurobiological baseline. Consequently, the brain becomes highly receptive to behaviors that force artificial dopamine releases. To learn more about how mood, stress, and wellness connect, visit co-occurring.ai. ACE, our AI chat explorer, provides a private, anonymous, and evidence-based educational experience. This concludes our overview of the biology of reward deficiency. Please refer to these sources for further information.